Well, hello. My name is Greg Harlock, and I am an assistant professor of neurosurgery here at the University of Utah. It's my great privilege to contribute the chapter Neurochemical Pathomechanisms and Traumatic Brain Injury to the very prestigious Yeoman's textbook. I need to, right off the bat, acknowledge the important contributions of my co-author, Dr. Christopher Zacko, who's a neurosurgeon at UPenn in Hershey. And both Dr. Zacko and I feel it's important to acknowledge Dr. Ross Bullock. He mentored the two of us through the previous version of this chapter, and he's been a very important mentor to the both of us throughout our careers. So we're indebted to him and appreciate his important influences in this chapter. Now, another issue of housekeeping is that I've additionally contributed the chapter on spinal cord injury, basic science, to the Yeoman's textbook, and that creates a little bit of an issue in terms of overlap. What I've tried to do with the two chapters is in the basic science behind head and spinal cord injury, there's been a bit of a different focus, and traditionally head injury has been more focused on neuroprotection, whereas spinal cord injury has been a little bit more focused on regeneration. And I've tried to use those as a basis for some distinction in the two chapters. But of course there's necessarily some overlap. I trust that you'll find these two chapters highly complementary, and they should be viewed as such. Now, in terms of head injury, so head injury is a very important public health problem, and despite decades of research, it continues to be the case. I think a very important point in discussing primary head injury is that we sometimes say that physicians are helpless to do anything about primary head injury, and when I think the reality is quite different. I think we need to remember that as physicians, we can play a very important role in patient education, public health advocacy, and also designing safety technologies, all of which can either help to prevent primary head injury or to reduce its devastating effects. So I hope that all of you keep this in mind as you're watching this. Now, one of the other things that I'd like to talk to you a little bit about is the issue of distinguishing secondary insults from secondary head injury. Secondary insults were recognized back in the 1980s as a result of the Traumatic Coma database, so a very important study. And I have to tip my hat to Randy Chestnut, who made the very important discovery that patients who had not only a head injury but coexisting hypoxia or hypotension had significantly worse outcomes. And this has given rise to the notion of secondary insults, which exacerbate the damaging effects of a head injury. So much of the work that we now do as clinicians is to reduce the incidence or severity of these secondary insults, or even better, to prevent them from happening. This is really all that we have to offer head injury patients really at the moment. And again, it's important to distinguish this from secondary injury processes. So I would now like to take some time to speak in more detail about these secondary injury processes. This really is the focus of the textbook chapter. So the different processes that we need to talk about include hypoxia, ischemia, edema, excitotoxicity, calcium dysregulation, cytoskeletal proteolysis, mitochondrial derangements, oxidative stress, inflammation, as well as cell death and apoptosis. So now a few introductory words about each of these topics. Hypoxia ischemia is a real problem after head injury, and what we've recognized is that it is seen in pathological specimens to a greater extent than would be expected as a result of hypotension or hypoxia. It's not really understood why that is. It may be related perhaps to exacerbating excitotoxicity. But the bottom line is that we're now focused in our clinical care in terms of trying to augment and optimize delivery of nutrients to the injured brain. We've learned that cerebral perfusion pressure therapy may be more important than intracranial pressure-directed therapy. And one of the experimental technologies that I've watched with interest has been the issue of perfluorocarbons. And so these are synthetic molecules that basically can augment the oxygen carrying capacity of the blood with the goal of optimizing oxygen delivery to the injured brain. One of the major problems with this approach is the fact that the mitochondria may not be able to use that supply of oxygen, but nonetheless it's an exciting approach that's still being tested in clinical and preclinical trials. Another very important problem, especially after severe head injuries, is that of edema. And we've actually learned quite a bit about edema over the years. This is one of the aspects of secondary injury that we actually are able to treat. Obviously we have mannitol decompression and other therapies. The Medical College of Virginia, in particular Tony Marmaru, made the important discovery that actually cytotoxic edema, that is the predominant form of edema after a head injury, is not vasogenic edema. And this is very important because corticosteroids, for instance Decadron that we would commonly give to brain tumor patients, would not be expected to work after a head injury because these are really only effective for vasogenic edema. And one of the really key points of this chapter is that I hope, on occasion we still hear clinical stories of patients being treated with corticosteroids for their head injuries, and the reality is that this has been shown very clearly in the crash trials to be harmful. So again, there's no rationale for using them, and in fact it's been proven very clearly to be harmful, so please do not give steroids to patients with head injuries. Moreover, even if you are one who's inclined to give steroids for spinal cord injury, please don't give steroids to those patients if they have a coexisting head injury. Another very important secondary injury process following head injury, similar to what's known to happen after stroke, is excitotoxicity. So this is the excessive release of excitatory amino acid neurotransmitters. This is a self-perpetuating process, and it's also calcium dependent. There have been a number of experimental agents aimed at reducing the effects of excitotoxicity. Unfortunately, neurocognitive side effects have really limited the utility of the agents tested to date. Hopefully in the future we will have more success with this. Calcium persistently comes up as being a very important player in various secondary injury processes. It's linked to excitotoxicity because the synaptic vesicle release is calcium dependent. As well, calcium is known to activate enzymes that create reactive oxygen species. Moreover, calcium triggers damaging intracellular enzymes like caspases and calpains. Calcium hasn't been effectively targeted, but it does deserve mention that there have been several HIT trials examining the efficacy of nimodipine after head injury. And it wouldn't be entirely true to say that nimodipine wasn't effective. It should be noted that in patients with traumatic subarachnoid hemorrhage, in particular males, in several subgroup analyses there was a benefit suggested. Again, this is not strong evidence, this is subgroup analysis, but I think it's noteworthy, and it's certainly my preference in patients that have a large volume of basal subarachnoid blood. Even if you've excluded an aneurysm, I think that there is enough evidence to support putting these patients on nimodipine. Cytoskeletal proteolysis, again with caspases and calpains, this has been a very challenging aspect of secondary injury to target to date. What we found is that there have been some small molecule inhibitors of these molecules that have been developed, but they haven't actually performed very well in animal models. They certainly aren't yet ready for clinical translation. I think the really important consequence of cytoskeletal proteolysis right now is the issue of biomarkers. So there are a lot of people that think we would really benefit from having a troponin for head injury, something where we could correlate the severity of the injury with the level of a certain marker. And with things like, for instance, spectrin breakdown products, one component of the cytoskeleton, I think that's most likely where we're going to see aspects of the cytoskeleton contributing to head injury in the near future. I think one of the key factors after head injury is the issue of mitochondrial dysfunction. And this is really an Achilles heel in the field because so many different experimental therapies aim to optimize nutrient delivery to the injured brain. But the reality is that if the brain can't use those nutrients, you're no further ahead. A very important discovery was the development of the so-called mitochondrial permeability transition pore. What this basically is it's a cyclophilin D dependent process by which mitochondria become abnormally permeable. They can actually go on to rupture, and this obviously impairs the electron transport chain and the supply of energy to the cell. This can go on to cause cell death. I think what's very interesting here is that we've actually found an experimental therapeutic that may potentially help with this. So it's been noted that cyclosporin binds to cyclophilin D and can actually inhibit formation of these pores. Obviously, one of the concerns with cyclosporin is that it's an immunosuppressant, but I think an important recent advance has been the discovery that we can separate cyclosporin's neuroprotective moiety from its immunosuppressive moiety. And there's a new agent that's currently in preclinical studies. It's known as NIM or NIM 811. And it's basically just the neuroprotective moiety. So far, preliminary findings are promising. I'll be very curious to watch this agent as it progresses through the various stages of preclinical and clinical testing. Oxidative stress is also very important after head injury. Reactive oxygen species are basically molecules with unpaired electrons in their outer shell, so they're highly reactive. They end up reacting with DNA, proteins, enzymes, cell membranes, and they can significantly disrupt cell functions and ultimately lead to cell death. Reactive oxygen species are largely produced as a result of the just discussed mitochondrial dysfunction. They can also result from, for instance, the inflammatory response and calcium-mediated inhibition of antioxidant enzymes. Antioxidants have been a very important aspect of head injury research in recent decades. Several antioxidants have been tested in human clinical trials, but none has been translated. In the instance of, for instance, PEG-SOD, this was a landmark trial which was unfortunately negative, and it's thought now in retrospect that it may have been negative because it didn't effectively penetrate into the injured brain. So still a very important area of ongoing research and thought to be a very important secondary injury mechanism. Neuroinflammation is also a very important mechanism of secondary injury. Neuroinflammation is a very tricky business, and that's because the inflammatory process after head injury is both beneficial as well as deleterious. And my personal views are that neuroinflammation may be a very tricky therapeutic target. That's not to say that we shouldn't try, but personally I'm banking on some other areas of research in terms of hopefully beneficial effects in head injury. And lastly is the issue of cell death and in particular apoptosis. Now, one of the big things that I've learned about head injury over the years is that cell death is a bit of a tricky business. Classically, we're taught that there's necrosis on one hand and then apoptosis on the other. Necrosis is this default energy-independent process, whereas apoptosis is the so-called programmed cell death, where cells in an energy-dependent process go through a somewhat slower process of dying. And this distinction is somewhat blurred, and the reality is that cell death is sufficiently complex that they've even got what's called the Nomenclature Committee on Cell Death. And the reason they exist is because there's thought to be a lot of gray area between some of these different cell death mechanisms. And in fact, there's actually 4 different mechanisms of cell death that have been described. Besides necrosis and apoptosis, we also now recognize autophagy and something called cornification. We won't touch on those here and only briefly in the chapter. Part of what's complex is that it's now thought that there may be a bit of a gradient between necrosis and apoptosis. They may not be, strictly speaking, mutually exclusive. This has led to some cute terms like necroapoptosis or aponecrosis. And the reality is that this is quite an important distinction. We do have some agents, for instance, minocycline and cyclosporin, that we believe may have a benefit in mitigating apoptosis. So if we can say conclusively that apoptosis is an important or dominant mechanism of cell death after head injury, then it may be worth pursuing as a therapeutic target. So I guess as some concluding thoughts, I think that the basic science of head and for that matter spinal cord injury is, although it's stumbled a bit, it's a very exciting area to be involved with. I think we are seeing important progress, and I think we've learned a lot from failures of the past. I would certainly encourage readers to seek out the impact studies. So this is a whole series of papers, and this was published by Andrew Moss and his colleagues, and it basically serves to highlight a lot of important problems with the clinical trials completed to date, problems with study design and study analysis, and I think it will be very important for this new generation of head injury researchers to bear the lessons of these studies in mind. So with that, I will close. Thank you very much for your time and your attention and for your interest in this chapter. If for any reason you have any questions about this content, I'm easy to find on the internet and would welcome an email from you. All the best.